ఇంటర్మీడియట్ విద్యారంగంలో అగ్రగామి మన NSR ఇంపల్స్ ఎడ్యుకేషన్ ఇన్స్టిట్యూషన్. ఐఐటి జేఈఈ అయినా నీట్ అయినా తిరుగులేని విద్యా సంస్థ NSR ఇంపల్స్.
Hello students, now let us continue with the concept of Chemiosmotic Hypothesis. Now Chemiosmotic Hypothesis, as we have just started for the last class. Now who is the scientist? Peter Michel. So this itself is called Peter Michel's Chemiosmotic Hypothesis. So when you take the Chemiosmotic Hypothesis, this is a hypothesis which explains about which phenomena? It explains about the process of ATP synthesis. It explains about the process of ATP synthesis. That means this is a hypothesis and theory has been given by Peter Michel. Of course, it's been studied and later established by Daniel Arnold, as we discussed for the last class. This is called Peter Michel's Chemiosmotic Hypothesis. Here, proton gradient concentration plays very important role. So that is a chemical reaction takes place that creates osmotic potential. And this is a theory, means imagination. This is how the process is going to happen, and this hypothesis itself is being widely accepted for the synthesis of knowing the process of synthesis of ATP. So this is what itself is called Peter Michel's Chemiosmotic Hypothesis. So this theory is for what? Process of ATP synthesis. In the process of ATP synthesis, if you look at, we say adenosine triphosphate. Always production of energy is being associated with phosphorus. So wherever we find phosphorus, we say that is a good source of energy, right? Wherever we find phosphorus, that is a good source of energy. Now what we are trying to synthesize? ATP. ATP means adenosine triphosphate. That is why this process itself is also called as, or itself is called as, phosphorylation. Itself is called as what? Phosphorylation. Now what we are going to study? We are going to study the process of ATP synthesis or phosphorylation. What is phosphorylation? Synthesis of phosphorus, synthesis of ATP itself is called as phosphorylation. Now this we are discussing. Means Chemiosmotic Hypothesis explains about ATP formation itself is called phosphorylation. Then next question, as I told you for the last class very clearly, the two important cell organelles plays very important vital role. What are they? We have chloroplast and mitochondria, right? So let us go in depth of this. So this phosphorylation occurs in two cell organelles. Just I introduced for the last concept for the last class. So which are the two cell organelles we have to study? Nothing but one is nothing but chloroplast, and very important. Now the point what I am giving you as a basic point, very important. Another cell organelle is nothing but what? Mitochondria. Very important. Right, so we know both of them are nothing but what? Both of them are called semi-autonomous cell organelles. Why they are called semi-autonomous cell organelles? Because they have their own DNA, and DNA has ability to undergo replication. So in supportive of this we have studied for the last class. So where the energy synthesis occurs, where ATP synthesis occurs, where phosphorylation occurs means semi-autonomous cell organelles, because they have their own DNA, it undergoes replication, and they are nothing but chloroplast and mitochondria. So we are discussing about phosphorylation. Phosphorylation occurs in two cell organelles, right? Now if you take in chloroplast, where is the location of phosphorylation? Location or site of phosphorylation. site or location of phosphorylation, meaning where the ATP synthesis takes place within the chloroplast, right. Then what do we say, that is nothing but granum thylakoid, or we say lumen of thylakoid. Okay, granum thylakoids, or we say lumen of thylakoid. Suppose in the question you get the site of phosphorylation in chloroplast or photophosphorylation if you take. If granum thylakoid option is not there, you have to choose the lumen of thylakoid. If this wasn't there, then you have to choose the option of granum thylakoid. This is a place where what happens, is a place where there is a synthesis of ATP. ATP is produced. How ATP is produced here? Now if you look at, this is a chloroplast, here you know this is thylakoid, one thylakoid membrane we are speaking about, onto which what is falling here? Light is falling. Photo means light. So light is very much essential. Which light? Sunlight is very much essential to break the water. We discussed for last class. Protons will accumulate. Using these protons, ATP is synthesized. So using light, there is a synthesis of ATP where in the lumen of thylakoid. That is why this is called as, right, photophosphorylation. Photophosphorylation. Okay, so this is called as what? Photophosphorylation. What is the meaning of photophosphorylation? Photophosphorylation means light is needed for the process of phosphorylation. How do you define here? The process of ATP synthesis in the chloroplast or in the lumen of thylakoid, in bracket write chloroplast, by using sunlight. This is total process of ATP synthesis. So how do you define photophosphorylation? What are the terminologies are important? ATP synthesis is nothing but what is taking place. Where it is taking place means in the lumen of thylakoid. Where the lumen occurs means within the chloroplast, and that is by using sunlight. So this is what we call it as photophosphorylation. Right. Now coming to mitochondria. In mitochondria, where the phosphorylation occurs, let us take the site or location. So again I am writing site of phosphorylation. Always you have to understand that whenever I write phosphorylation, always you have to remember in your mind, phosphorylation meaning synthesis of ATP, synthesis of energy, right. So this occurs in inner membrane space, mostly this you will get it as in the form of question, and this also the site or location, mostly you will get this as the questions. People always have the confusion with these two parameters. The site of phosphorylation in mitochondria means you have to remember inner membrane space. The site of phosphorylation in chloroplast means you have to take granum thylakoid or lumen of thylakoid. Right. Now if you take inner membrane space here, now as this is called as, this process is called as oxidative phosphorylation. Oxidative phosphorylation. Why? Here this is called photo because light is needed for the synthesis of ATP within the chloroplast, that is why it is called photophosphorylation. Then why do we call in mitochondria, why do you call ATP synthesis as oxidative phosphorylation? Here it is because of the food. What is the food produced in plants? it is sucrose, glucose or maybe starch, right? That is being broken down, the glucose or the starch or whatever you take, sucrose, which is produced as a result of photosynthesis. As a result of photosynthesis, food is broken down, meaning food undergoes oxidation. Food undergoes oxidation, reacts with oxygen, right, and lead to formation of carbon dioxide, formation of carbon dioxide, water, along with that energy is released. What is that energy? Itself is nothing but ATP. Okay, so here in mitochondria also ATP synthesis occurs. Where it occurs means inner membrane space. What do you call that? Means oxidative phosphorylation. Why it is called oxidative here? Here it produces ATP with the oxidation of food. By breakdown of the food, energy is released out. That energy is stored where? In mitochondria, right? So here how do you define this? The same thing you can write here. It's like the process of ATP synthesis, right, where in inner membrane space, this may visible, may not visible, so better I will write on other corner. I will write here process of ATP synthesis, where in inner membrane of mitochondria by oxidation of food materials, itself is called as, it's called as what? Oxidative phosphorylation. Why? Because look at here, we are speaking about phosphorylation, that is nothing but ATP synthesis. Where is happening means site is very important, inner membrane space of where, which is cell organelle? Mitochondria. And how? By oxidation of food materials, itself is called oxidative phosphorylation, right. Before I raise the board, I will tell you this part is very important. Okay, again I will try to represent in other way of remembering few things here, but anyway, Peter Michel's is called Chemiosmotic Hypothesis. I will give you quick revision, concentrate. Process of ATP synthesis itself is called phosphorylation. It mainly happens in two cell organelles. Here these two are called semi-autonomous cell organelles because they have their own DNA which undergo replication. We studied for the last class cell division, karyokinesis, cytokinesis. They undergo replication at the time of cytokinesis. See this, these concepts are related to first year cell division, right. Now chloroplast, where in chloroplast the ATP synthesis occurs means granum thylakoid, lumen thylakoid, very important question. Now it is called photophosphorylation. What is the definition you have to remember? Write down so clearly that the process of ATP synthesis in the lumen of thylakoid of chloroplast by using sunlight. Chloroplast synthesizes ATP. Photo means light, that is why it is called photophosphorylation. Quickly coming to other corner, in mitochondria where it happens means inner membrane space, and the process is said to be oxidative phosphorylation. Here food is getting broken down, that glucose or sucrose what is produced in the photosynthesis, it undergoes oxidation, reacts with oxygen and produces carbon dioxide and water, along with that ATP synthesis occurs. So here it can be defined it as oxidation of food, that means process of ATP synthesis in inner membrane space of mitochondria by the oxidation of food materials is called oxidative phosphorylation. Same thing when you have to write, make sure that you write and remember. Yeah. So let us write this. Phosphorylation, how many types? Phosphorylation, it is of two types. Again I am giving you a good version, that is very important, because this is a place where most of the students will have a confusion. Let us write very clearly. Phosphorylation, it is of two types. What are the two categories? Photophosphorylation, right? Yes, using sunlight. So where it occurs? It occurs within the chloroplast. Sometimes indirectly they will ask you the question, when photophosphorylation takes place means, so it occurs in during photosynthesis. So photosynthesis is very important to remember. It happens during photosynthesis. What you can remember? First one, photophosphorylation occurs in chloroplast during photosynthesis, very important. And the second category, it is called as oxidative phosphorylation. Right, phosphorylation is of two types. Second one is oxidative phosphorylation. Where it occurs? It occurs in mitochondria. Now in which process it occurs? It occurs during respiration, very important. It occurs during respiration. So sometimes they will understand the whole concept. Most of the students, they will understand the whole concept, they will read the whole concept, but they will not connect the point to photosynthesis. Photophosphorylation occurs during photosynthesis. Now we are in the process of discussion about in the chapter called photosynthesis. And one more thing, here how photophosphorylation occurs? It occurs through electron transport system. Where it occurs means, in which stage it happens means electron transport system. Remember one more very important difference. Each and everything what I am writing on the board, each and everything, each and every bit, each and every part, each and every point, each and every box what I am giving in the form of a block diagram, each and everything is very important to answer the questions on photosynthesis. Guys, so concentrate. If you take where photophosphorylation occurs, definitely by passing electron from one substance to other substance. At the stage, which is stage? During electron transport system. So if you take in case of photosynthesis, we have two electron transport system. I am giving you clear difference. Two types. What are the two types? Guys, you know it very well. Non-cyclic electron transport system. Other one is called as cyclic electron transport system. See, everything is interconnected. In non-cyclic, what we discuss? The synthesis of ATP as well as NADPH. During cyclic, what we discuss? Synthesis of only ATP. Now, coming to respiration, in respiration also what occurs is, where ATP synthesizes means definitely no doubt by ETS only, means electron transport system only. But here we have only one electron transport system, that comes where? After Krebs cycle. After Krebs cycle, here it happens in ETS takes place. ETS takes place in where? Inner membrane space of mitochondria. Very important. Meaning there is only one type of ETS in case of respiration. But in case of photosynthesis, there are two types of respiration. That means photophosphorylation includes two types of ETS, two types of electron transport system. But oxidative phosphorylation includes only one type of ETS. See if you can clearly make note of each and every basic point. Guys, nothing can confuse you on solving any kind of question. Let it come from any corner of the textbook. Let it folded and twisted number of times, make it as a tough question, but still no problem. You guys can answer very easily, provided if you are thorough with the concept, right? So now this part is very important. So as I told you, phosphorylation is of photophosphorylation, where it occurs means chloroplast during photosynthesis. Here two types are there. What are they? Non-cyclic electron transport, where both two energy sources are produced, ATP and NADPH, whereas in cyclic it is only ATP. Coming to oxidative phosphorylation, it occurs in mitochondria, and during respiration, here ETS occurs after Krebs cycle. ETS takes place in the inner membrane space. That means here two types of ETS, here only one type of ETS. And one more thing, you know it very well, this is mitochondria. Regarding mitochondria, when you take energy storehouse of energy, or you know storehouse of ATP, whenever you get the question storehouse of ATP, right, or sometimes you get the question that ATP furnace, right, storehouse of ATP or ATP furnace, or itself there is one more name for mitochondria. Can you people recollect it for first year cytology? Mitochondria is also called as, I'll just arise and write. There is one more terminology which you need to remember from first year cytology. Mitochondria is also called as energy currency of cell. Energy currency of cell means for cell, who will provide the energy? Always it is mitochondria. So this all terminologies are related to whom? All these terminologies are related to mitochondria. Okay, mitochondria itself is called storehouse of ATP. Guys, you have to be very careful with the question. So people should not confuse with this. Sometimes there is a possibility of getting questions like this. Suppose if the question is storehouse of energy, of course if another question is storehouse of ATP, see most of the people will confuse, they think they both are same. But whenever the storehouse of energy question is there, guys, you have to remember biomolecules, first year chapter. Storehouse of energy is nothing but starch. Answer will be starch, not mitochondria. Guys, I always tell you, you need to have the keep each and every word carefully, you analyze on the question paper. See the each and every word carefully, you have to read the question. According to the question, you have to take the answer. Storehouse of energy, which act as storehouse of energy? At the time you have to choose starch as an answer, not as mitochondria, no. If at all the question is storehouse of ATP, then only you have to choose the option as mitochondria. Small difference, right? ATP is also energy, no doubt, but energy always comes from starch. And where it is stored means in the form of ATP. In the form of ATP form of availability, we will get it only from mitochondria. Okay, so who act as energy currency of cell? Mitochondria. Which is act as ATP furnace? Again the answer is mitochondria. Which is called storehouse of energies? Mitochondria. Similarly for the chloroplast related questions, what will be the questions on the chloroplast? So sometimes you will get kitchen of the cell. So here it's congested here, let me give you questions. So what are the questions you will get it on chloroplast? Kitchen of the cell, or we call producer of cell, or sugar factory, which act as sugar factories. For all this, which act as sugar factories, for all these questions, what is the answer? You have to remember the option as chloroplast, not the mitochondria. Chloroplast, not the mitochondria. So which act as kitchen of the cell, meaning where do we prepare the food? In the kitchen. Similarly, food is prepared by the chloroplast by photosynthesis. Producer of the cell, it produces starch. Starch acts as storehouse of energy. It produces starch, means it produces a source for production of energy, that is a starch. Right, that is why it is also called producer of the cell. And what food is prepared here? Sugar. What sugars? Sucrose, starch, glucose, fructose, mannose, galactose, these all are called sugars. So, they are called sugar factories. Which act as a sugar factory if the question? Producer of the cell or kitchen of the cell means you have to take the chloroplast. So, according to the question you have to choose the right option. Let us continue, guys. Guys, here you have to be very careful with the questions related to phosphorylation. This is quite simple if you practice it, but remember you have to see the question carefully before taking the option. You have to see whether the question is speaking about photophosphorylation, you have to see whether the question is speaking about oxidative phosphorylation. So, here finally what we have to discuss, Peter Michel's Chemiosmotic Hypothesis. Okay. Now, listen carefully. So, to study the chemiosmotic hypothesis, it requires, what it requires? This is also mostly you will get it as in the form of question. Mostly you will get the question that Peter's Chemi-Michel's osmotic hypothesis. To study Peter Michel's chemiosmotic hypothesis, what are required in the process of ATP synthesis. Okay, because this hypothesis explains what? This hypothesis explains finally about what? This explains about phosphorylation. Phosphorylation meaning what? ATP synthesis. Right now. So, this is mostly we get it as in the form of question. So, remember it requires 4 components. It requires 4 components. What are they? Try to say carefully. It requires a membrane. It requires a proton, a concentration gradient, it requires a proton pump and an enzyme ATP synthetase. What is ATP synthetase? It is an enzyme. So, it requires 4 components. Okay. First of all, it requires the membrane. It requires a membrane, and membrane need to have the proton concentration gradient. You need to have more concentration of the protons into that membrane. Then it requires some pump to transfer the protons out of it, and also requires an enzyme called ATP synthetase. And these 4 components are common for both chloroplast as well as for mitochondria at the time of ATP synthesis or during the process of phosphorylation. Now, here why protons are there? Because if you take, protons are needed for ATP synthesis. To produce the ATP, the enzyme required is ATP synthetase. right. Now let us, before we start with the osmotic hypothesis here, very important means here mostly we are discussing, now we are in the chapter called photosynthesis. So what we are discussing here, photophosphorylation. So you know it very well, where it occurs? It occurs in the chloroplast. So when you look at the here, very importantly, where it occurs in non-cyclic electron transport system and also during a cyclic electron transport system. So who is responsible here? What are the things apart from the four are very much required. Apart from this, what are the things you have to remember here? Photosystem 2, a quinone cycle and a photosystem 1. Right, and where the entire story is happening? Entire story is happening in the lumen of thylakoids. And from where the protons are coming? Protons are coming from the stroma. Why? Because PS1 is located in the stroma. The total, sometimes they will give you that identify, we have a question. We have the question, identify the site of locations of the chloroplast which are related to chemiosmotic hypothesis. So at that time generally what people will do, you know, blunder mistake at the time, they will give this as options as A, B, C, D, E and they will ask you to find out first option is all A, B, C, D, E, second option B and D. Most of the times when they ask you to choose, people will skip PS1, people will skip stroma. They think that PS1 and stroma are not related to photophosphorylation. Guys, you have to remember photophosphorylation also occurs in cyclic, In cyclic who is involved? PS1 is involved. So you have to choose even option PS1 because PS1 also generates ATP, and where the PS1 is there, what you call thylakoid oppressed thylakoid of stroma or thylakoid oppressed region of stroma. What is thylakoid oppressed region of stroma? I told you for the last class, right. So here that and this PS1 exists in stroma. Generally they will ask me after the exam and they will come and tell me, generally in weekend exams also, they say that ma'am, stroma is not involved in non-cyclic. No, because stroma is only giving the protons. Stroma is not involved in non-cyclic. No, then how you have to choose including stroma? How we have to choose stroma? Remember guys, stroma is giving the protons. Stroma is a contributor of what? Stroma is giving the protons. Proton contributors we have two. Who contributes proton? So when you take proton sources, whenever the question is proton sources means from where we get protons, what are the sources to get protons means we have two sources. One is water, that is splitting of water, other one is stroma. If stroma wasn't there, stroma is not there, protons how do we get it? Because that is why we say what are the components required for phosphorylation means protons are needed, proton concentration is needed, to pump the protons even pump system is needed, means protons are very much essential. See here, if protons are not there, if stroma is not there, protons cannot come. If protons are not there, then there is no ATP synthesis and no phosphorylation. So guys, generally when the question is given, so the structures, the components needed for photophosphorylation in chloroplast means people will choose PS2, they will choose phenol cycle, they will choose these three and they leave PS1 and stroma. Guys, make sure that because not only non-cyclic, cyclic also involved in ATP synthesis, you need to choose even PS1 as well as stroma also. I am telling you again, remember this, where what are the things needed for photophosphorylation in the chloroplast means you have to take the components of both non-cyclic as well as cyclic. This question, next time if you get this coming weekends, if you get the question, make sure that you have to remember, or in mostly this question comes in the grand test. In the grand test, they will study for the entire second year, they will do some 2 times, 3 times, 5 times revision of the chapter, but when the question appears in the grand test, people will confuse and they leave certain things which are very much needed. If these two wasn't there, there are no protons, because proton contributors are two. Splitting of water that takes place in the lumen of thylakoid, other one is stroma. So compulsory you have to include PS1 and stroma. So guys, I will just quickly tell you, this question also mostly appears in the grand test, like after you complete the revision of all chapters. So Chemiosmotic hypothesis, it requires 4 components guys. If you have these 4 in your hand, wherever you need, you can synthesize ATP. Of course, in living organism, right? What are they? A membrane is needed to transfer, to pump the protons, then proton concentration gradient is needed. From where you are getting protons? Water and stroma. A proton pump is needed, some pump system to push the protons out of the membrane, then ATP synthesis enzyme, right. Now, we are talking about now photophosphorylation, not the oxidative phosphorylation. Oxidative phosphorylation we are going to study in the respiration. People generally what they will do, you know, students, they will read photosynthesis, they will understand photophosphorylation. In the next chapter, they will read respiration. At that time, they study mitochondria. At that time, they study oxidative phosphorylation. But if the question is related to both chloroplast and mitochondria, both photosynthesis and respiration, they will feel tough to answer the interrelated questions. So you need to have comparison. We are discussing here photophosphorylation, not oxidative phosphorylation. Oxidative phosphorylation related to mitochondria, that we are going to study in next chapter. Respiration, at that time also we are going to study Chemiosmotic hypothesis, but that part also have the same introduction. Up to here everything will be same even in the next chapter called respiration. So now this is purely for photosynthesis. In photosynthesis what happens? Photophosphorylation, 2 types of electron transport system, non-cyclic and cyclic. So which components are involved in Chemiosmotic hypothesis? Means PS2, Phenone cycle, PS1. Up to here non-cyclic. Of course, it occurs in lumen of thylakoid. Make sure that not to avoid even stroma, because stroma and PS1 place in cyclic and they are nothing but contributors of the protons. So this is nothing but what we call Chemiosmotic hypothesis introduction. We will just start with Chemiosmotic hypothesis. Right. So if you take the Chemiosmotic hypothesis here, now we talk about chloroplast. So this is what is chloroplast. In the chloroplast if you take, we are going to talk about, these are nothing but thylakoids. These are nothing but what? Thylakoids. Now we are taking only one thylakoid. We are going to take about one thylakoid. So I am taking this thylakoid out. This is total chloroplast and here light falls onto this, and this is the thylakoid membrane, and inside what you find? Lumen. This is nothing but lumen. Right, and from which what happens? There is a synthesis of ATP. Each thylakoid itself is involved in ATP. Of course, total granum. In granum means what? Piles of coins of arrangement of thylakoid. Each thylakoid there is a synthesis of ATP. So to understand Chemiosmotic hypothesis, what we have to do? I am taking one thylakoid out. So we will study the total story in one thylakoid. So what I am doing? I am taking the thylakoid out. So this is chloroplast. Now what I have to draw now? I will draw now a thylakoid. What do you call this? This is called as a thylakoid. I am talking about one thylakoid. Innerly what you find here? This is nothing but lumen. What is that? Lumen. Now look at here. From here this is PS2. This is PS2, and next is it enters into cytochrome B6, cytochrome F complex, and what you find here is nothing but PS1. Let us imagine that is PS1, right. So when you take this is PS1 here, now what happens here? From the PS2 it comes here and it goes again up PS1. What do you call this? This is called Z scheme. Z scheme meaning non-cyclic electron transport system. So from the PS2, when electron is coming, first it enters into what? First it enters into the component called as Pheophytine. First it enters into the component called Pheophytine, non-cyclic electron transport system. What is Pheophytine? Pheophytine itself. What we have to remember about pheophytine? Pheophytine is nothing but what? A primary electron acceptor of PS2. A primary electron acceptor of PS2. That means from the PS2, first who will accept the electron? Pheophytine. Where the pheophytine is located? Pheophytine, site I am writing, or the location, site or location. Sometimes you will get the question that where pheophytine is located. The site or location of pheophytine, remember, on the outer membrane of thylakoid. Outer membrane of thylakoid. What do we say? Outer membrane of thylakoid. Pheophytine not on the inner membrane of thylakoid. This is thylakoid. What you find here, this is thylakoid. What you occur here is thylakoid membrane. So, whenever the question you get the question, the site or location of pheophytine means the location of pheophytine is outer membrane of thylakoid. What is a pheophytine? A primary electron acceptor of PS2, right? Then it enters into where? It enters into a plastoquinone. So, what it occurs here, it is called a quinone cycle. It occurs quinone cycle. I will use red marker to show very clearly how the phenomena occurs for the next class, right? Quinone cycle, it gets operated with the quinone cycle, right? And plastoquinone. Plastoquinone located where? On the thylakoid membrane. Try to remember, plastoquinone is located on the thylakoid membrane. Who is located outside the thylakoid membrane? That's nothing but pheophytine, right? Quinone cycle, the electron comes to cytochrome B6F complex, plastocyanin goes on to PS1. So, in this plastoquinone, each electron what is carrying, one electron carry 2 protons. So, it drops the one electron, what it drops is nothing but 2 protons, right? So, altogether in non-cyclic electron transport system, I will just give you note. Right, here I will just try to give you the note. Try to remember the note point here. Non-cyclic electron transport system. Take it under note point, very important. Sometimes if you get the question that total number of protons participating a non-cyclic electron transport system, how many total number of electrons? How many you will get it? 1 electron 2 protons, and second electron 2 protons 4 protons. From the stroma 2 protons 6 protons, and in NADPH 2 protons, right? 8 protons. Total number of protons are 8. Out of 8, stroma contributes how many protons? 4 protons. Why stroma contributes 4 protons? 2 protons it gives for ATP synthesis, and 2 more protons it will give for NADP synthesis. Very important, need to recollect it. Total number of protons involved in non-cyclic is 8. Out of that, stroma will give 4 protons. 2 protons are for ATP synthesis, 2 protons are for NADP synthesis. That means in the stroma proton concentration decreases. Proton concentration decreases. Guys, what I told you last point, try to remember. With this we will continue for the chemiosmotic hypothesis.